Hello guys, my name is Dr. Varun and you are watching Chlorophyll classes only on YouTube. Meanwhile, let's come back to our 12th class session and we have started what molecular basis of inheritance. Quickly, not wasting any point of a time, I just want to recapitulate what we have done so far. We were talking about that we have what inheritance is through the molecules. Okay, we have studying it through the molecular level and we have studied something called a DNA and we were studying about it. So we studied a lot of about it and we're gonna be taking it to the next portion or the topic. Along with that, in the last video we were saying that we have studied about that the DNA look something like this, certain values are there, and we have even studied about that Chargaff's rule. Now in this video we're gonna be seeing what Chargaff's rules application would be and how we can use it. I hope you know that what is Chargaff's rule. If not, go back and see the last portion of the topic. From that video you will get it. Let's just, as I promised, we will be solving certain questions on it. So these are the questions we're gonna be doing it up. These are the very basic questions, not a deeper level questions. So we will consider it as LOD level 1 type of questions from the topics which we have studied last. So here, if you see that they are saying that we have what, a double stranded DNA. In this, the amount of guanine is what, a nucleotide, okay, is 35%. Then what would be the amount of cytosine? Now we know that in our DNA, what is this is a DNA will have what, it's a double stranded structure, right? It will have a opposite pair which will be pairing with it, because DNA is always double stranded, remember it better. Only the RNA single stranded is present with us, okay, in the viruses. Now if you see that they are saying guanine is 37%, okay, G is 37, means its counterpart would also be there, right? G will always make a bond with C. We know that from that Watson and Crick model as well as from the Chargaff's rule. So we know guanine makes bond with cytosine. So means whatever be the amount of guanine, same would be the amount of cytosine, sorry. So guanine amount will be equals to cytosine amount, right? So how much is guanine? Guanine is given 35%. So this is 35%. So this would be what? C also 35%. So answer would be option D. Okay. Now the next question is over here and I want you first to try yourself and pause this video over here and then I will let you know explain it. So if you have tried it, okay, then I'm gonna help you out in this again. So they are saying that in a DNA of an animal, percentage of adenine is 30% is adenine. We have to find guanine. In the last, what we know that particular thing will make a bond with the particular one. What we know that adenine will make bond with the thymine, whereas guanine will make bond with the cytosine. Now what is given? This is given. What has to found? We have to find about this. Okay, in the last question which was easy, we know that opposite would work, but here what will work? The questions just become little longer, nothing else. Let's see what is happening. So this total would be what? Total would be 100%, right? 100% would be made up of these nucleotides only. Now, if we know about this first, like I'm going to use this one. So if we know about the adenine, we can get the thymine, right? So adenine is this. So adenine is what? 30%. Thymine will also be equal to it, that is 30%. Means how much total? Total nucleotide, that is 60% is this only. So the left out will be G plus C, because total is 100, 60 is done, left out would be this. So that would be what? That is 100% minus 60, that is 40. So they will be 40%, G plus C. Now both are same, G will be equals to C. So can we say that if we write it 2G is equals to 40%? So what I will do, G value will be 40 minus 2, that is 20. Do we have an option? Yes, we have an option. This is the way you will write it in your academic exams. But this much is not needed when you will be calculating in your academic NEET examination. But I try to give you the answer of it. Let's see another type of questions, which is not from the Chagas rule, but from the structure of DNA. Now, in the last video, I have told you that it is like double helical structures moving like it. But how long? That is the major question, that how long it would be. On that basis only we are going to do one question on it, and then we will see the problem with it. Okay. So let's just see this one question. What the question is saying? It's saying that the distance between two consecutive base pair is 0.34 nanometer. We already know that between two base pair we have this. Okay, if you remember this thing, we said na, this between these two base pair the distance is what? This nanometer. We have even done it in the Armstrong. Okay, can come to you in your examination. Okay. And the total number of base pairs in the DNA double helical of a typical mammalian cell is 6.6 into 10 raised to power 9 base pair. Means just like a human diploid cell, this is being, they have given that this number of base pair is present in the DNA. You know that DNA is what? In the form of a thread-like thing, right? So then the length of the DNA. So how long this is? Like we have tell multiple of this, right? And we even know what is the distance between them. So if we multiply both the thing, we can easily get the distance of it, because it is what, like you get this number of this, right? And this distance, you know it. If you 1, 2, 3, 4, these are what multiple base pairs that is present. So you can easily tell what is the distance, like what is the number of, what it would be the approximate length of the DNA, right? It's very easy. So what we have to do, we have to just simply multiply this thing with this thing. Okay, so 0.34 nanometer, okay, or we can simply write it like that. Nanometer can be changed into 10 raised to power minus 9. Nanometer means minus 9. Multiply by 6.6 into 10 raised to power 9. Now over here, this thing and this thing will be cut. What we will have to 0.34 into 6.6. So 66 into 34. That is 6 4s are 24, 26, 18, 18, 19, 4, 4, 9, 10, 11, 12. Okay, and we have 3 dots, 1, 2, 3. So that is 2.2 meters. We have changed it to the meter, 10 raised to power. This is what changed into what? Meter. Okay, so this is the answer. Now the question is, what is 2.2 meter? 1 meter is from my this hand to this part. This is almost 1 meter. 2 meter will be doubled. Now you'll be like, sir, how is it even possible that we have 2.2 meter long DNA segment? Then the question arising after this is that, okay, if it is true, okay, which we have done it, okay, we are not bluffing with the question, okay, it is 2 meters, 1 meter or more than this distance is there, total length is there, then how it is packed inside one single cell? Because each cell, my body is made up of single cells. Okay, now in each cell, how is it possible that it is packed, that 2.2 meter or 2 meter long DNA is packed inside each cell? So here I give you the example of how the DNA packaging is done. So just think about, you all went to tailor. If you have seen a tailor, or at home also your mothers might have a simple swing in thread. So if you know that the thread is coiled around that cylindrical cardboard structure, okay, and if you open that thread, the thread is so long, means what? If we coil the things, it might get what? Condensed, or means it come up in a very small space. This thing happens over here also, and this is only we're gonna learn about it. So we're gonna find out who gonna be acting the cylinder for the thread of our DNA. Okay, understood this thing? Able to imagine it? Let's just see it. Okay, so the next thing that we're going to be studying is the packaging of DNA. Packaging of DNA. Now, you know that where DNA is present. DNA is present inside the nucleus of the cell. So if I have to make a nucleus of the cell, so I'm going to make that. Okay. These are what? These are the endoplasmic reticulum which are attached to the nucleus membrane. Okay. Now on this nucleus, we know what? We know that there is a presence of what? There is a, okay. There is a presence of what? There is a presence of ribosomes. Okay. Because till now, if you remember the 11th class, you might know that the ribosomes are formed by the nucleolus. Nucleolus is what? It is a, like when we stain the nucleus, it is a darkly stained area, and it is what it is doing, this making the ribosomes. So this membrane, like the nuclear membrane, it is having what? Lot of ribosomes present, which are even extending it to the endoplasmic reticulum. Now if you see over here now, what I said, that genetic material will be present inside this nucleus. So I'm going to be writing it up. These are what? This is my nucleus. Now inside nucleus we have our genetic material. Now the genetic material is present in the form of threads, and we call them what? We call that chromatin. Okay, so nucleus material is in the form of thread-like structure like this. How much it will be? 2.2 meters, right? So this is what? This is your chromatin. Nucleus thread. Now, if you will take this thread, how this thread will look like, let me just show you. Like this. Oh, what happened? Okay. So this thread, it is in the form of double helical like this. And there will be what? These are what? Double helical structure. There will be what? Base pairing that would be present. So if I had to show it, I will show it like this. Okay. Okay, let's make it a little better. The other strand I'm going to be showing with a darker color, but it's not visible. Wait. Then I'm going to be showing it with a white color. Okay, so it has what? It has base pairs present. So these are what? These are the base pairs which I'm showing with the red color. Okay, this is your genetic material, what we have studied. Now this thread has to be coiled. Okay, how it will be coiled? It will be coiled with the help of a protein called as histone proteins, which are present in eukaryotic cell. Here I have to remember it, very important, that eukaryotic packaging. Because in prokaryotes, beta, there is nothing histone protein present. There is a non-histone proteins which are working in the packaging. Okay, so this difference also we will remember it. So now what's gonna happen? This thing has to be coiled around a structure, means our thread or the cylinder of the thread, which we remember. So here it is what? It's a histone protein, where this, like, imagine that this is the one. Okay, this is nothing. Okay, so this is the one. Here what happened? The thread will coil, like thread what? This DNA. Okay, this will coil like this and goes on, and then again there will come, again it will coil. So multiple time it is coiling around this histone protein, not just single but multiple of them. Okay, so we're gonna be writing about it. Okay, so if you see, there is what? These are the histone proteins. Okay. Now, this is our DNA. What happened with the DNA? Oh, this is our DNA. Okay, so I just keep it like a lot of things are coming up and you are able to understand. It coiled around it. Okay, so it will, as it will coil, what would the coiling look like? Something like this. Like this. Can you see that there is a coiling like this happening? So it is being coiled like this. So the yellow color thing is what? It is the histone protein. So this is what? Histone protein. Now, we know that this is what? This is our DNA helix. Okay, we're going to be writing those facts also over here. So what are the facts? The facts goes something like this. So the first thing is what? This is what? Your DNA double helix DNA. All the features will be present over here. Okay, so this is what this is called as chromatin, DNA genetic material, which is a chromatin. Now this histone protein and DNA, why they are planned over here? Because over here they are being attracted towards each other. Two reasons. Number one reason is, if you remember when we were making the phosphate is what? Negatively charged. Okay, and this histone protein is positively charged. First thing. Okay, so this negative and positive will make what? A electrostatic bonding. Positive negative will be attracted, so electrostatic bonding will be formed. Second thing, which is also confirming that they will be coming together because of the acid basic nature. Okay, your DNA is what? Acidic in nature because of phosphate present into it. Your histone protein is basic in nature because of presence of basic proteins which are positively charged. Okay, what are those basic proteins? Lysine and arginine. Now you will say that how these proteins are positively charged. If you remember about the concept of Zwitter ions, that plays into it. If you don't know about the Zwitter ions, let me know in the comment section. I will help you out with the shorts. If you want to know about it in biomolecules chapter when we're going to do it, you can go back and see the amino acid topic of it. Clear? We understood few things about the histone proteins and all those. Let's just see this bonding over here. So protein, this we're going to be writing up over here. That is protein associated with DNA are positively charged, which are what? Histone. Okay, in eukaryotes. In prokaryotes, they are less positive charged non-histone protein, which makes, this is for both, which makes a electrostatic bond with negatively charged due to what? Due to phosphate DNA. Just a minute. Now let's just see about the histone proteins. We have already told that it is a basic in nature, having arginine and lysine. More about it. Actually, this histone globule that you are seeing, that circular structure you are seeing, actually it is what? There are total 5 types of histone proteins. And in this histone protein, which is rounded about it, it has only 4 types. Last 5th type is present somewhere else. Let's see that where it is present and what are the 4 present inside this globule area. So the first histone protein is present over here, and it is act like what? It is act like what? A stabilizer. Okay, between that, if you see that there are 2 globule and in between the DNA is flowing. Now think that how delicate it is. So to stabilize that area, this H1 histone protein is present. So H1 histone protein is present over here. This area is termed as linker DNA. That part of DNA which is linking between the 2 histone proteins or the adjacent histone proteins. Okay, and this histone protein, like where I'm talking about this, it has what? It is a new body. Why we call it new body? Because it's an octomer. Okay, so it is what? It looks something like this if I have to make it. Okay, this is so it has what? It has H2A protein, H2B histone protein, H3 protein and H4 protein. That too in the double H. Okay, means there are total what? 2 of this. Okay, there is what? All of them in the 2 forms. Okay, means they are what? Doublet. So this is what this area will be called as what? Octamer. Histone protein octamer, because 8 of the histone protein are present in it. Now this structure is called as new body. Okay, you will remember it. And this new, like this one histone protein, very important these terminologies. This one histone protein with DNA is called as what? This structure is called as nucleosome. What is nucleosome? DNA wrapped around the histone protein. How much wrapping is there? 200 base pairs. Okay, this 200 base pair is wrapped around it, which takes how much turn? 1 3/4. Means one full run and the other will be 75%. Somewhere it is 65% also. Okay, not complete 2 turns. So this nucleosome, if I have to write, nucleosome is what? It consists of 200 base pair DNA chromatin plus new body. This is what your nucleosome. How much it is wrapped? Turns or wrapped DNA will be 1 3/4. Sometimes it is written as 1.65. Okay, 1.75 also according to the fractions, but this is how we write it. Now let's come that what this nucleosome plus linker DNA is called as. Okay, so this terminology is also very important. So this is called as chromatosome, because here all the type of histone proteins are present, included, because H1 protein is also included in it. Okay, so the linker DNA plus nucleosome is called as chromatosome. Remember it. Now what's gonna happen? Okay, let's see. Now these nucleosomes and linker DNA, what happen? Means this chromatosomes, they come more closely together in the group of 6 and form a structure called as solenoid, because now these things are wrapped more like a solenoid. Solenoid is what? It is what? A magnet wrapped by a electric wire, copper wire or something. Same thing, they are also wrapped, coiled, means highly congestion is there. Able to imagine how it will happen? Like this. And in this, when you see that this one thing, okay, can you see this one rule? In this, there is, these are individual of them. Okay, in each individual of them, 6 nucleosomes are present, beta. Means 6 these things are present and coiled over here. Can you see that how much coiled this DNA would have become like this, like a solenoid? Okay, so that thing will happen, and that is only I'm gonna make now. Okay, so making it from the rough. So like this. So finally it will be what? It will be highly congested solenoid kind of structure. Okay, so I'm gonna be making it like that. Okay, so let's just complete it. So this would be what? And the blue color means the second strand. Okay, so this area which I have made over here was called what? It is called as, this is called as solenoid. Now how much millimeter diameter? Okay, one thing over here I forgot to overtell, that this area which we have made, the first one over here, this has a dimension of around 11 nanometer. So means original whatever the size of DNA, it is reduced to what? 1/3. Reduced to 1/3 of original. Okay. Meanwhile, this solenoid, this is 30 nanometers. Further, it is being reduced. Okay. So this area is 30 nanometer. Now what happened? They will further condense and form what? This yellow structure which I have made, that is what? Super solenoid. Okay. So this area is what? It is super solenoid. And this is how much small? 300 nanometer. Think about it, the packaging that has occurred. Now this super solenoid kind of a thing is present till your normal DNA, but more than this can also be present, and that happens during the time of cell division, and it forms structures like chromatids and the chromosomes. Okay, so what gonna happen? They will further condensed. Okay, so I'm gonna be making it with the lighter color, or maybe a darker sheet. Okay, so what will happen? These things will further condensed. Okay, so this thing is like this, and further condensation is there. This structure which I have made, which is further smaller than the ones which we are making, this structure is what? This is the chromatid, which is 700 nanometer. Okay, this is 700 nanometer, and this will be seen during the time of, this will be seen during the time of cell division. And you can observe these things when? During the metaphase. Okay, metaphase, you can observe these things. And the distance between these two means this and this distance is 250 nanometer. Okay, now what happened, this each 700, like chromatids are what, these are the parts, these are what, the sister chromatids of the chromosome. So finally it goes and forms something like what, which I'm going to be showing up to you, that is okay. So this structure is what, this is the chromosome. Understood? So this structure will finally be forming what, these things. So chromatids, if you know about the chromosomes, this chromosome has two chromatids here and here. Okay, so what formed chromosomes, which is having a dimension of what, this both strands, making it 1400 nanometer. This level of constriction or compaction occurs. Now the question is that, sir, why this much of compaction is actually being taken place? Because the size is getting reduced, so they have to compact it more. And during this cell division time, more than the non-histone proteins come into it. And what we call them, non-histone chromosomal proteins. Okay. To enable further compaction, certain non-histone chromosomal proteins. This is what, non-histone chromosomal protein are needed to form chromatids and chromosomes. Okay, so let's just see this whole thing. If you see that initially you will see the chromatins. Chromatin is what? DNA. DNA will first coiled around the new body, which is made up of histone proteins, and that body is called as nucleosomes. Nucleosomes, multiple of them will join together and form a solenoid, which is 30 nanometer, further compact. These solenoid will change further into super solenoid, that is 300 nanometers. This is what the maximum is present in the normal cell. But during the cell division, they will further compact with the help of non-histone chromosomal proteins. And they will compact further to the 700 nanometers, which is what, chromatid structures. And chromatid is the part of chromosomes. So one chromosome has two chromatid during the cell division times, right? So this will be what, 1400 nanometer of compaction of the genetic material. Understood? So this you have to take care. This whole thing I have made it, go through it, see it. These notes will be available on my Telegram channel as well as on my WhatsApp channel as well. The links would be there, so you can get these notes from there to study and to do the rest of the things. So this is what the packaging of DNA look like, which is one of the super important thing. So if someone will ask you that, write what will form after what, so we will remember it. So first you have what, you have your double-stranded DNA, which is 2 nanometer. Okay, then it will form into what, it will form into nucleosome, and nucleosome was what, 11 nanometer. Somewhere it is written as 10 also, so 10 to 11 nanometer. Then this nucleosome is forming what, solenoid. So solenoid is what, it is 30 nanometer. Then it will form super solenoid. This super solenoid will be what, 300 nanometer. Okay, moving on to the next. During the time of cell division, they will form chromatids. Chromatid is what, beta, 700 nanometers. And finally, you have chromosomes, which is 1400 nanometer. This is it. Now the question is, okay, we had made a packaging of DNA, but how this DNA is actually working? Okay, very good questions. That, okay, DNA has all the information, like for example, each cell has the DNA. How each cell is influencing the height? Major question, that giving me this height, how that is possible? Is this DNA is telling that, okay, the DNA which is present here, it has something and it will say that go increase height. It never happens like DNA doing it. So DNA works in a hierarchical method. Means like in your school, you have what? You have a principal, then head teachers, then teachers, right? So principal will tell that I want the result to be like this. It will come to the head teacher. Then head teacher will tell to every teacher that bio teacher do this, and we need this result. You get the result and the teacher work and give you the results, right? This happens like this. Same thing, DNA also, like if what my body is saying that I want to give this much of height, then the DNA was like, okay, I will give this information to the RNA. Okay, like I have the thing, I will give it to the RNA. Okay, RNA said, okay, fine. Then RNA will go and form the proteins. Okay, means RNA is what? Head teacher, DNA is what? Principal, and protein is your teacher, which will do its effect, which will give the height. So what we learned from here, that in your body, all the things that is being done with the help of proteins. Means if proteins are there, you are able to talk like this. If the protein are there, I am able to do this. If the protein is there, you are able to understand the things which you are seeing over here. So we understand that proteins are simple and most important thing in the body. And we can't get, you say, we are eating proteins, so we will get the proteins. No. For your body has to make a particular proteins. So why we are eating the protein? Major question comes. If that is your question, I would like to take your little importance over there. Okay, so let me throw some light on you. Okay, don't worry, I'm not breaking the light and throwing on it you, just giving you the things about that, what happens. When you eat a protein in your diet, what happens? It go and get digested. That means it is not going to your any cells, right? So it is being digested. Why it is being digested? Because inside the protein, there is a presence of amino acids. Because if you remember little bit from 11th class, if not also, I am letting you know over here that the proteins are made up of amino acids. Amino acids, few are formed inside our body, few we need it from our diet. What diet? Amino acid is not a diet. Okay, the diet is what? The diet is the proteins which are made up of amino acids. Understood? So we are eating protein so that we can get the amino acids which we need for our protein forming. Clear? Interdependent everywhere. So this information, how it will flow of the genetic information is known as central dogma. Okay, so we're going to be studying the next thing is what? Central dogma of information. What it states? It states that the information flows in a certain way. Okay, that is flow of genetic information. Okay, so from DNA, it goes to RNA. From RNA, it goes to protein. Now, DNA to RNA, the process of forming from DNA to RNA, we will learn it later, don't worry, don't get confused with a lot of things, but RNA is a genetic material. No, it has other functions also, which we're going to be learning over here. So DNA is what? DNA's function would be RNA, sorry, to make the RNA is called as transcription. And RNA to protein, it is called as translation. Okay, from transcription and translation. So we have learned this thing. Now, we understand the body function like this, but DNA also need to be keep on forming in the new cells so that new cells can keep on doing this function, of course. And DNA will be formed how? DNA will be formed by DNA itself. Okay. So this is called as replication. So in our syllabus in the next video, we would be studying about these steps individually: replication, transcription, and translation. But this is what central dogma of information, how the information is being done. But there is a one problem with certain organisms, like certain viruses. They do what? There is a presence of reverse central dogma. What is reverse central dogma? Okay, so reverse central dogma. So it happens in what? In certain viruses. Which viruses? Like your HIV virus, tobacco mosaic virus. Basically, they are what? RNA viruses. What happened in them? Okay, now what happened in them is that normally they have what? They have RNA present. They do what? They go back and they form DNA. How they form DNA? Till now we have said what? DNA will make RNA only, not the back forth, but here it goes back forth. So what happened? The RNA in those viruses where RNA acting as a genetic materials, but they will infect the DNA carrying organisms. What they will do? They will from the RNA will make the DNA, and this process is called as reverse transcriptase or transcription. And for this enzyme, reverse transcriptase is present. So when we're going to be studying about the HIV virus life cycle, there this thing will come. Very important for you to understand what is happening. Certain viruses have RNA as their genetic material. They change itself into the DNA and then they make the proteins. Okay, so this process, so now when they did it, what happened? They make the viral DNA, it will go make the RNA, and then the proteins will form. This is what? Reverse transcriptase, reverse flow of genetic information, in which happen RNA containing viruses. Example, HIV virus, tobacco mosaic virus. 11th class tobacco mosaic virus, here HIV virus. Okay, other viruses are also there, but we are not studying. This is what information flow and how we can be studying the next topic that we're going to be completing over here. That is the search for the genetic material. We said that there are two types of genetic material, RNA, DNA, right? How did we know that this DNA will be playing as a genetic material? Why not carbohydrates? Why not proteins? This thing has been always a question of big curiosity amongst the scientists. So scientists were working continuously to find out which gonna be the possible genetic material. Let's just see that. So the next topic that we're gonna be studying is the search for genetic material. Search for genetic material. Now there were three different experiments that we have to remember. Number one experiment is the Griffiths transformation experiment. Okay, so the first experiment is what? It is transformation by Frederick Griffith. Okay, when he did it, 1928. Okay, now he did what? He tried to find out what is something which can be giving a certain features. We know that genetic materials gives the features, and he took what? He took a variety of bacteria. What are the varieties of bacteria or the strains of bacteria? Let's just try to understand. So he used a bacteria. So I'm gonna making a bacteria. Okay, not good enough. So this is one bacteria. Okay, imagine it. And he took the two strains of the bacteria. Okay, so when I'm saying the two strains, means two different types of bacteria. Okay, so one bacteria appears rough, other appears smooth. Okay, so this is smooth bacteria because it has what? It has a coating present around it, whereas this is what? This is the rough bacteria. Now these are what? He took the strains of the bacteria, two different strains. So the rough strain, I call it R2 strain, and the smooth strain I call it smooth 3 strain. Okay, now strain of what? There are pneumonia causing bacteria. Okay, now who will cause the disease? Smooth one will cause the disease because the rough one do not have an immunity against the my body defense system. Okay, so means when the rough will go inside my body, it will be find out by my body immune system and it will kill it. Tishkam. But when it comes to smooth one has a coat around itself which gives us what certain properties to prevent my body to identify and kill it. Clear? Let's just see this, which bacteria it is which will cause pneumonia. And you know that pneumonia is deadly if not treated. Okay, so what will happen? Which bacteria strains we're gonna be doing? We're gonna be either taking the Diplococcus pneumoniae, or it is Pneumococcus pneumonia, or Streptococcus pneumonia, or Haemophilus, whatever it is. Okay, we're going to be taking those bacteria. Okay, so we have taken this one. Now we need a guinea pig. So who we will do experiment on? All the mice, mouse, rats. We do all the experiment on them, right? So let's just do the experiment. Just think that I am putting we are taking these strains into the different injections. I am the graffiti. Now we will become graffiti and we will see that. And we have a mice. Okay, so we took the first mice. Okay, the first time mice. So mice. Okay, so this is our first mice. Okay, so this first mice which I have taken, I have put the rough one into it. Okay, the first mice, and I give it what? I give it rough one. Rough one. Okay, rough is injected into it. What will happen? Rough will be able to be found by the mice immune system and it will kill it. So mice will survive, means no disease. Okay, so no symptoms of the disease will come. No symptoms, and it will survive. Okay, now the second case. Okay, in the second case, what I did? I took the mice. And this time I put in his body the smooth one. Okay, now everyone knows that what will happen when the smooth will go? Smooth will not be detected by it. Symptoms will come, means it will get pneumonia like symptoms and it will die. Because I'm not giving any medicine. I am doing what? I am doing the experiment. So I learned that there is a one strain which has just one thing different. That is the outer coat, which is giving a specific category. Now, this outer coat is made up of protein, right? So everyone thinks that this protein is the genetic material. Okay, let's just not go over there, but I'm giving you the briefing of it. Let's do one more thing. What we did, I heat killed this smooth one. Okay, so in the next experiment, what I did, I took the mice, third experiment. The mice is turning more like cat now, but this is mice. Okay, hatta katta mice. Hatta katta means what? Fatty. Okay, fit mice. And I give this bacteria only, but I heat killed it. Heat killed it means what? That I put this into the heat. Just think about it, that if I put you into the oven, what will happen to you? Sir, I will die, right? You will say this thing only. So this bacteria will also die. So a dead bacteria would have what? Like this eye. So this is what? Dead. Okay, now when I put this heat killed bacteria, heat killed S strain, when I put into it, then what happened? No symptoms comes. No symptom comes and the mice survive because you killed it bacteria, na? So it won't. Now what it does, now I'm a graffiti, I did one more experiment. This time I take this two where no symptoms came, means this is alive, this is dead, and I put it, and then what will happen? Our observation should be what? It will come what? It will come survive, no symptoms would come. Let's see that what happened. Okay, so in the fourth experiment, I took the mice and I added what? I added a live rough one and a heat killed the S1, and this is heat killed. So this is what? This. So alive R strain and heat killed S strain. And what happens? Symptoms occur. Okay, this is something different which was not expected by it. And it also see what? It died. Mice died. Okay, then we did what? Then to find out what has happened to it, I took this dead mice and extract fluid from it, because pneumonia has what? The fluid present, and analyzed it. And when I analyzed it, I found what? I found live S strain present. And I was like, what is this? I have put the killed S strain. How come they becomes live? We know that life doesn't get itself, right? We know that like if someone is killed, no one can come back, except it's some movies or serials. Then the people can even come back after getting death, right? But here they don't. That's not possible in real life, right? That is a brain rot which is being fed to you. So what will happen? This life S strain, how it comes up? So the things were given, like there were certain materials which were present in the heat killed S strain, which is uptaken by the R strain, and it evolved or transformed itself into what? S strain. What material it would have taken to transform itself? That is not the capsule, but the genetic materials which will be making that capsule and change this R strain to the S strain. So what conclusion was given to it? Conclusion: R strain took up certain material which is called as what? Genetic material from heat killed S strain and transform itself into live S strain bacteria. By this we come to know that there is a genetic material which can change the composition of any other organisms. So we understand this. See this once again and you will understand it pretty well. Let's just start with the next one. This is the first experiment, and we don't consider it. Till now we didn't say that it's a DNA. We call it's a protein. Okay, because protein is showing all the effects, right? So we said that protein is a genetic material. But no, later we have to see that whether it's a protein, it's a carbohydrates, DNA, RNA, what type of molecule it is, because we are studying, our chapter name is molecular basis of inheritance, right? Let's just see. So next experiment that come up, that was what? That was biochemical assay. That is biochemical characteristic of transforming principle. So means the transformation has been done, and what is the biochemical that is doing it, means whether it's a DNA, it's a RNA, it's a protein, what it is. Okay, transforming principle. Now this experiment is being done by three different people in 1944. Okay, so 1944. This was 1928. Years are important in this chapter. Name of the scientist is very important in this chapter. Okay, let's just see that. So there was what? Avery, MacLeod, and McCarthy. Now they extracted the molecules of protein, carbohydrates, RNA, DNA from the S strains. Okay, so they did what? Extracted molecules like protein, lipids, carbohydrates, etc. from S strain, because we know that these any of these molecules, whatever these molecules are there, they would be responsible for the transformation of the R strain. So we have already taken out the molecule from the S strain. So I am making what? An S strain over here. And I am the person who is taking out what? Molecule from it. Okay, so I have taken out different molecule. So what are the molecule? Number one, I have taken the protein out, extracted either all the proteins which is present, or there would be what? RNA into it in its body, or DNA, all the other material, but I am doing with this three only. Other answer would be similar. Okay, now when I take it out and I want to put it, like I want to see that which will be uptaken by the S strain to change itself. So what I did, I take the test tube. I took the test tube, okay, and I take three different test tubes. Okay, here only I'm making. Now in these three test tubes, what I did, I put the R strain into it. Okay, so here I put what? In this test tube, R strain is there. Okay, it will be big. Okay, multiple R strain is present inside it. Understood? So what is here? Test tube A, B, C. Okay, have R strain. Have R strain. In all of them has R strain only. Okay, R strain. Okay, now what I did, I took one by one the extracted thing from the S strain and put into it and see that whether they will change or not. So in the first this, I put protein of S strain. Then I put RNA of S strain. Then I put wait, let's go back. Put extract from S strain. Just extract only I am putting. Put extract all of them from S strain. Put extract from S strain. Okay, I put it all the extract. So I am making it like what? This is the extract. Now the next thing is I'm gonna be putting the enzymes which will digest it. So for example, protein will be digested by protease. Okay, so what I will put now? I will put the protease. What the protease will do? Protease will break all the protein particles. Means the protein particle is not present in the A1. Okay, so what will happen, beta? Will break all the protein, right, of the extract. Okay, I did it with the extract only. I am telling you over here. Means no protein left. No protein left, right? The second one, I added what? I added RNAs. RNAs is what, beta? It is that it will break all the RNA present from the extract. Okay, will break all the RNAs present in extract of S strain. Means no RNA left. And the last one, I would be adding what? DNAs, which will break all the DNA present inside the S strain extract. Okay, so what will happen? Will break all the DNA present in extract, means no DNA left. Now let's see that whether they will change or not. Okay, so in this R strain, what is present? There is a present. So what happened in this? There is a presence of RNA plus DNA. Whereas in this B, there is a presence of what? Protein plus DNA. Whereas in the last, there is a presence of protein plus RNA. Now, in the first one, if we see that, we found what? We found there is a presence of what? There is a presence of S strain. S strain appeared. Means that if I delete all the protein also, it is getting transformed. Means that protein is not the genetic material which is changing it. It will be either RNA or DNA. Proved protein isn't genetic material. Did you understand this? Means if the protein, people were thinking that the protein would be the culprit behind it. If I delete the protein and still the same crime is being done, means of transformation, means protein is not the one, the other thing might be the one. Cool. Then the second one, that what? Protein is destroyed. Like, let's see that protein is taken out. Like, protein is not the one. We are only left with RNA or DNA. Let's try with the RNA. When we put the RNA, like RNAs, and RNA is got away, still we found what? We found that there is live S strain. Means transformation is occur. S strain appeared here also. Means here what thing is not present? What is the thing? RNA was not there. Protein is already taken out. Means we already know that protein is not the one also. RNA is also not the one because we have destroyed it. Still, what is left? DNA. Let's just confirm it by the final also. And by now we have already understood that it's a DNA. Okay. So it proved that what? This thing proved that RNA isn't genetic material. It isn't genetic material which is doing this transformation. So let's see the last one. In the last one, we destroyed the DNA of the S strain. Now we didn't found any S strain. So what happened? There is R strain only. Means there is a no transformation occur. Okay, means no transformation. Which means what? Means DNA is the one which is the genetic material. It is being proved by the two experiments A and B test tube also, and confirmed with the third one. Proved that DNA is genetic material required for the transformation. Go through it, see it, and let me know that. All these notes are available on my Telegram channel. Link is in the description, as well as it is present on my WhatsApp channel as well. Its link is also there. So this is how this experiment is being done. I try to make it as easier and less complicated to you. And we come up with a third experiment, which is a confirmatory experiment. Then people will say that why not this is acting as confirmatory? Because at that time, people were not very sure or confident or agreed to this experiment style. other experimental style is being done by the Hershey and Chase. To understand that, you need to know that what is transduction. Transduction is the method by which virus infects the bacteria. And what it does, virus attaches with the bacteria, releases its genetic material inside the bacteria, leave the outer area of the viruses. And if you don't know what is virus, we know that virus is made up of two things only, proteins and the genetic materials. So we know that either the protein is culprit, or the genetic material, or the DNA or RNA, whatever they are present, that will be the genetic material. So this is how we will be able to find it out. Let's just see that. So the last experiment is, the third experiment is in 1952 it occurs, that is by the Hershey and Chase experiment. This is very important experiment because this is coming again and again in your board examination. For NEET, everything is important. For board, that is simply very important, this one. Okay, so it occurs in 1952. Okay, now transduction, you need to know what is transduction. Transduction is the method of reproduction in which bacteriophage is what? Bacteria disease causing virus is called as bacteriophage. So bacteriophage infects or transfer genetic material into bacteria. Okay, now we know that bacteria is made up of only two things, protein or the DNA. So we have to find out whether the DNA is the genetic material or the protein is the genetic material. Here we will be able to do it. What we did a radioactive technique. We make a virus which is radioactive in terms of its protein, one virus, and other virus is radioactive with respect to its DNA. Okay, now which thing we made is radioactive? In protein, there is the presence of sulfur, so we make that radioactive. In DNA, there is the presence of phosphorus, not sulfur. No sulfur is being seen in case of DNA, right? So we made the phosphorus as the radioactive. Now, what is the property of radioactive? You can see the radioactivity on the autoradiography or in the radioactive films, where it will easily illuminate and you will able to find out, okay, this is the radioactive substance. Okay, so this thing we're gonna be doing. So we made two different bacteriophage. One bacteriophage, this is the first one. So this is what the first bacteriophage, its genetic material is radioactive. So I made it with the green color. Whereas in the second one, its outer coat means your protein coat, which is what? Outer coat, like this. This is radioactive. But inside this is. So whatever the thing bacteria will release inside, that will be the genetic material. Now, whether it will release this or this, that depends. Okay, means whatever bacteriophage, sorry, not the bacteria, bacteriophage, okay. Whatever the bacteriophage will release inside the bacteria will be genetic material, right? Easy peasy. Let's make a bacteria. So this is your one bacteria, and this is another bacteria. Okay, now what happened? The first step would be what? It's infection. So in infection, what happened? It go and attach to it. So first step would be what? Infection. So infection, what happened? This bacteria and this bacteria both attached itself to it. After infection, what will happen? That we will, they will transfer the things. So what things would be transferred? The genetic material. So this will transfer this will also transfer this. So what will happen? Inside this is bacteria. this goes inside, and here the genetic material of the second one goes inside. This is what? This is your bacteria. Bacteria. Now we will blend it. Blending is only what? Mixing it up. Okay, so now this thing happened, and then we will centrifuge. Centrifuge means what? We will shake it up so that the bond will break up. So when we will shake it up and the bond will be broken up, what will happen? See, this is the bacteria, first bacteria. This is the second bacteria. Okay, the outside area is called as supernatant. This is what? Outside area. Outside area will be called as supernatant. Okay. Okay. Now, in the first bacteria, what will happen? This DNA will go and incorporate into the DNA of the bacteria. Whereas in the second one, what will happen? Same thing will happen. The bacteria, it will get incorporated in the DNA of it. Outside there will be what? There will be present a protein coat only, because outside it is there. Here what? Protein coat, because after centrifuge what will happen? Like just think that someone is making you go ringa and just leave it. So what will happen? You will go outside from that area. You won't be keep on stuck with it. Basic physics. Okay. Rotational motion, right? Here also we are doing the same. So if you see the outside area and the inside, you will find the radioactivity where? Here in the first case, radioactivity observed inside cell, whereas here radioactivity observed outside, that is in supernatant area. Means if I am the bacteria, the outside this whole area, whatever is not with my body, that is a supernatant. Okay, supernatant area, which tells us what? I told you that. Remember this fact that whatever we are released inside will be the genetic material. What is released? The first bacteria's DNA is okay. So DNA becomes what? The genetic material, and it is proved over here. Okay, hence DNA is genetic material, proved over here. Right? So now the question is that what we did over here, we made what? Phosphorus radioactive. Whereas here we make sulfur radioactive. That's it. You have to remember it, what is happening where. With this, our quest to find what will be the genetic material is over. Now let's see that why not RNA become the genetic material? Why DNA only becomes the genetic material? Okay, so the question is that if I had to tell you that there's a difference between DNA and RNA, which you can check it out in the shorts video. Here I'm going to be simply telling you why not RNA. Look, in this world, when initially the life started to form, we will learn in the evolution chapter, RNA was formed first. Later on DNA will form, because DNA is what? It is a changed or evolved form of RNA, right? Why it happened? There might be certain shortcomings with the RNA. So what are they? Number one is that chemically RNA is not stable. It is highly reactive and keep on changing. How you will remember it? Remember that coronavirus came, COVID came, right? That COVID or the Wuhan virus, whatever it is, that is our RNA virus. And we said that, okay, the new strain has been found, this strain is more deadly. Remember these news were there everywhere. Why there's a new strains coming up? Because of RNA present into it. Because of RNA, they keep on changing very fast. Mutation happens very fast. They're not stable enough. Okay, so this is the one reason not stable. Why they are not stable? There could be one reason that their sugar is ribose sugar, which is chemically less stable. Okay, they have another difference, that is there is the presence of a easier, simpler compound with a less number of functional group, that is uracil, compared to DNA, which has what? Thymine, which has a methyl present over it, which is making it more stable. Moving on, deoxyribose sugar, which is what? More less reactive. Okay, because H is there. OH is more reactive, which can react and form some new things. Hydroxyl group. Not remember, go to the first page, first part of this video and see that what is happening there. Okay, with the structure thing. So we understand this thing that the RNA is a less stable, chemically less stable, more reactive. That's why we need what? We need the DNA that is being formed, which is more chemically stable. Understood? Now the next thing, that is, so what is the properties of genetic material? Okay, so the question which I gave you, what? Why RNA did not come up as genetic material so the answer is due to its less chemically stable nature. And you know that how this happens. You will write these notes. Along with that, for a board exam, there is a question called DNA versus RNA that you also have to do. If you want me to do, I will make a short out of it. Coming up to the major question, then what are the properties of genetic material? Properties of genetic material. Don't worry, this is the last topic. Okay, genetic material. So the properties of genetic material is, number one, it should be chemically stable. Okay, first thing, should be chemically stable. But now the question is, should it be 100% stable? No, it should be stable that slow and steady changes are present there. Okay, means that it should not be so stable that if the environment changes and we are not able to change our genetic material, means we won't be able to survive. That's why evolution is needed. And that happens because slowly DNA keep on changing, not an instant change. Understood this thing? Let's just write this up. Should, must be mutable. Okay, must be mutable with, must be mutable but show positive slow change due course of time to contribute in evolution. Next thing is, it should able to maintain its integrity and replicate. If something can't replicate from its own self, then it's not be able to do anything, right? Should able to replicate. Should able to replicate. Okay, and that's it. It able to maintain its structure and all those things. Yeah, able to maintain its structure and properties throughout the generation. Okay, throughout generation. That's it. Well, that's all for this video. Thank you so much for watching till this. If you like this video, give it a like, share it with your friends. Don't forget to subscribe if you haven't yet. You can see my rest of the videos which will help you out. We are trying to complete the NEET 2025 this year in a good and a nice way for the students. Well, that's all for this video. Thank you.